Now that we've touched on some practical examples, moving on, we're going to do a deep dive on what makes a good experiment. During these next couple of days to weeks, you are going to work on designing your own experiment before coming to your first in-person lab. So it is very important that you pay close attention to the material coming up in this unit. Sound good? We want to make sure that you are set up for success. So with that, let's get started. So diving into what makes a good experiment. First things first, a good experiment should provide data to support or refute hypotheses. Just as another reminder, we never prove anything in science. We can disprove things, but we work to provide support for theories and hypotheses. That is the purpose of an experiment. So remembering the purpose is the first step in designing a good experiment. When designing your experiment, it is ideal to only test one variable. If you test more than one variable at a time, you might get results that can't conclusively say whether your experiment actually elicited the results that you are interpreting. So looking at one thing at a time is very important. Another thing that we all have to be cautious of, as we're all human, is working hard to remain unbiased. This means that we need to make sure that we do not let our opinion influence our experiment. It's also very important that we repeat our experiment, so that way we can ensure that our results are not just what we call a one-off, right? We have to make sure that our experimental results, I should say, did not occur just by random chance, but because of what we are testing. So repeatability is very important. And it's also very important that we work to remove confounding variables or external factors that may influence our experiment, right? Such as the environment, the time of day, the equipment being used. It's very important that we standardize certain things in our experiment. So another way to say this is consider and eliminate all confounding variables. The term confounding variables just refers to variables that can influence your experiment that you don't want to influence your experiment, or that maybe you didn't think of. Sound good? And then speaking of variables, a good experiment should always have very clearly defined independent, dependent, and standardized variables. And then we also need to define our groups. There should be a clearly defined treatment group in our experiment, as well as control groups, with an adequate, meaning large, sample size. Remember, sample sizes are very important. That kind of ties back a little bit to repeatability, making sure that our data is trustworthy. And then speaking of data and results. It's important that these are analyzed appropriately using statistical analyses. Otherwise, how can we trust the results that we got? Without proper analysis, there's no way to say whether or not our results mean something, or if we are just getting those results due to random chance. Okay, so this is our list of things that you should keep in mind when you are developing your experiment. We're going to break down some of these now together to make sure that you understand what is needed and that you are set up for success, okay? So let's start out with types of variables. In a good experiment, there are three types of variables. We have our independent, dependent, and standardized variables. Starting with the independent variable. The independent variable is kind of what it sounds like. It's the variable that is not dependent on any other variable in your experiment, right? And so this is something, the data that stands alone. It isn't changed by other variables that you are trying to measure. Sometimes that means that the independent variable is what's manipulated by the researcher, okay? And every experiment should only have one independent variable, ideally, because you should only be looking at one thing at a time in a well-designed experiment, okay? Moving on to dependent variables. Dependent variables are what you hypothesize to change when the independent variable changes. Right? This makes sense. This is where it gets its name from. A dependent variable is dependent on the independent variable. It's what is observed and measured. It's how you collect your data, or what you collect your data from. Sound good? Something that's really important to note, though, about both of your independent and dependent variables is that you measure your units very carefully and that you label what units you used when taking note of your methods in your study as well as your results. So when you're reporting on your experiment as well as designing your experiment, noting how things are measured and the units that you used, such as grams versus meters versus liters, is very, very important. And now finally, last but not least, are standardized variables. These poor guys often get overlooked, but they are so important, just as important as a dependent or independent variable, because without standardized variables, your experiment might not elicit proper results. Standardized variables are the variables that researchers standardize or keep the exact same in all of their groups, right? So making sure that we are controlling for certain things so that way we're only testing the relationship between the independent and dependent variables. The reason why standardized variables are so important is because they help avoid confounding variables. Remember, we really only want one thing to be different between each of our experimental groups, and so that thing is usually the independent variable, what we are studying, the treatment. This is why having standardized variables is so important. So with that out of the way, let's go ahead and look at an example to make sure we understand. Charles' law states that as temperature increases, the volume of gas expands, and conversely, as temperature decreases, the volume of gas decreases. You design an experiment to explore Charles' law. What would be the independent variable? What would be the dependent? And then what would be the standardized variables? Pause the video, take some time. Okay, now checking back in, what would be the independent variable? In this example, our independent variable would be temperature, because temperature is not related to anything or dependent on anything else in this experimental design. In fact, the volume of gas is dependent on the temperature. So off the bat, we know that temperature, which we are increasing, right, or decreasing as part of our experiment, so it's manipulated by the researcher, the volume of gas is dependent on temperature. So temperature is our independent variable, and volume of gas is our dependent variable. And then standardized variables, there are a lot of options for this. One of them could maybe be the apparatus that you used, making sure that you're using the same equipment across the board when you're repeating your experiment and gathering more results, more data points. Maybe the amount of sunlight that's exposed. You never know if something is in sunlight, maybe it's swaying your temperature a little more than you would have thought. Or measurement devices used, making sure that you're measuring using the same things across the board to make sure that you're avoiding those confounding variables. Just a couple examples. Sound good? Okay, let's move on to your next check-in. Check-in number 5. Jamie is conducting an experiment to help answer the research question, does age impact weight loss? Jamie gathers a sample size of 100 voluntary participants of varying ages for their experiment. To minimize confounding variables in their study, Jamie has all participants live in the same type of housing, follow the same diet plan, and all undergo the same exercise regimen for 1 month. From this information, determine what is or are the independent variables in Jamie's experiment. What about dependent variables? And what are the standardized variables? Moving on to check-in number 6, making sure that this is number 6 that you are looking at. Is anything ever proven in science? So that's looking at variables. Let's go ahead and move on to looking now at groups. Within any experiment, there are 2 main types of groups, our treatment group and our control groups. Treatment groups are the groups or the group that receives the treatment. All right, so it is treated usually by our independent variable. And then your control groups are a point of comparison for the treatment group. So both of these type of groups in your experiment are incredibly important. The control group helps show what happens if the independent variable is not changed or added. And so, as I said before, this serves as a perfect point of comparison so you can ensure that your data collected from your treatment groups actually mean something. If your treatment group gets the same results as your control group, that won't tell you very much, right? Other than maybe your treatment doesn't work. So, this is very important. Now, within control groups, though, there are 2 kinds of controls. Well, 2 main kinds of controls, I should say. And those are our positive and our negative controls. Keep in mind that not all experiments have control groups, but most definitely should. All of the experiments that you are running in Life 102 Biology should probably have control groups, okay? So now let's break down the difference between negative and positive control groups. Starting with our negative control groups. Negative control groups are basically a group in an experiment that is set up the exact same as all other groups, right? So it's set up the same as your treatment group, except it receives no treatment. So the only difference between your negative control group and your treatment group is that your treatment group receives the treatment that you're testing, and the negative control group does not receive the treatment. It either doesn't receive anything, or it receives what we call a placebo. The purpose of a negative control is to serve as a baseline or a point of comparison for your treatment group. That's the negative control. And every experiment that you design in Life 102 should have at least a negative control. Now, a positive control, we don't always find, but it is always best practice. A positive control is a group that uses a different treatment, a treatment that is already known to produce a specific result. So again, this is the same as your treatment group, except instead of using the treatment that you're testing, you use a treatment that you already know produces a specific result. Why would you do this? Well, to make sure that your experiment is set up properly and to make sure that the equipment that you're using in your experiment is functioning, right? If you run your positive control and you get poor results or results that you weren't expecting, even though you used a treatment that we should know the result of already, that tells you that we can't trust our data from our treatment group because our positive control has told us that something has gone awry in our experimental setup, whether that's how we prepared our experiment or the equipment that we used in the experiment. Make sense? Okay, let's go ahead and apply this now with check-in number 7. Okay, keeping in mind, this is number 7. Dr. Nakia wants to find out if their research group's new cancer drug, Triumph, is effective. So they design an experiment to test the new drug. Participants are divided into 3 groups for their experiment. Group 1 receives drug A, which is already known to be effective at treating cancer. Group 2 receives Triumph, the new drug being tested. And Group 3 receives sugar pills, which should not have any impact on cancer cells. Based on this information, which group is the treatment group? Which is the negative control group? And which is the positive control group? If that problem was challenging for you in any way, or if you're just having trouble distinguishing and understanding the point and purpose of different groups in an experiment, no worries whatsoever. Please always feel free to reach out for assistance. And I have links to very helpful readings and videos already posted in Canvas, so feel free to reference those as well. Check-in number 8. Are standardized variables and control groups the same thing? Keep in mind, this is not a trick question. The reason I actually ask this question now is because some people in science refer to standardized variables as control variables, and this can make things very confusing, right? Because a control variable is very different than a control group. So I'm going to ask that you please avoid referring to standardized variables as control variables because it can cause a lot of confusion. We will use the term standardized variable in this course. Remember, a control group is very, very different from a standardized or control variable, okay? Awesome. Now that we have variables and groups under our belts, let's go ahead and start talking about data and results. We already went over how there is power in numbers, meaning the larger our sample size, the stronger or more relevant our data is, right? But something that a lot of students struggle with is how to display or share their data with others. It is always super important that when we share our results, that the information that we are sharing is actually, well, informative. Data should always be accurately recorded in a detailed data table whenever possible. But that information is largely for you and your research team. You then use this data table to create an informative figure. It is so critical that results we share with others is easy to digest and understand. The reason so many fear science is simply due to a lack of understanding. So it is incredibly important that we, as scientists, make a significant effort to ensure that our results and information is easy to understand. So, what do I mean by this? Let's pretend you are collecting data to see if blue fish have longer fins than orange fish on average. You gather a large sample size of fish and record their color and then measure their fin lengths. You then record your data in a data table like this. Okay, great, you have the data, but what do these numbers actually mean? On average, do blue fish have longer fins than orange fish? Your team decides to visualize your data with a bar graph. 2 graphs were drawn up. The graph on top was made by your coworker Alex. Alex made this graph by plotting each individual point from your data table onto the graph. For example, this first bar represents the first blue fish noted in your data table. And this last orange bar represents the last orange fish in your data table. In contrast, Joe drew up this second graph shown below. Joe made this graph by taking the average fin length for all blue fish and plotting it on the graph, as well as the average fin length for all orange fish, and then again plotting it on the graph. Now, let's circle back to your original research question and evaluate these graphs. Let's consider your response to the following questions for your answers for check-in number 9. Now, just looking at these two graphs, which one, off the bat, do you find more appealing or easily digestible? Which graph better answers your research question? Which one paints a clear picture and allows you to take a large amount of data and summarize it for people? This one, right? The graph made by Alex on the left is simply just a reworking of our data table. Both are very detailed and neither really paints a clear image to easily answer our research question. But Joe's graph on the right synthesizes the information from our data table, as opposed to just repeating it. It provides us with a clear comparison of fin length between blue fish and orange fish. This is why, when you are making a figure, it is always very important to use metrics such as averages, percentages, or frequencies. Now, we have 2 more topics relating to data and results, but you get to choose in what order you would like to learn them in. Consider this our official transition from learning about experimental design to now learning more about analyzing results and drawing conclusions. From here, we can learn about how we can determine whether our results are reliable or if they are just due to random chance. Or we can learn about why both Alex and Joe knew to make a bar graph for this dataset as opposed to a line graph. You need to learn about both of these before the end of this unit in order to be able to adequately analyze your results from your own experiment effectively, but you can choose in the order that you would like to learn them in. Sound good? Okay, with that, best of luck!